Hi, I'm Dr. Ramesh Menon, Director R&D, Personal Genomics and Genomic Medicine Divisions, MedGenome Labs. As today's World Parkinson's Day, so would like to give some insights on Parkinson's disease genetics. Broadly, we can divide Parkinson's disease, from a genetic perspective, it can be divided into familial Parkinson's disease or a sporadic Parkinson's disease. So familial form means, first degree or second degree blood relatives have Parkinson and, somebody is getting Parkinson's disease. And sporadic means it's a random, so no positive family history. So somebody gets a Parkinson's disease just without any family history. Now the majority of the cases that we see, 80 to 85% are sporadic. That means without any known family history. So and the total genetic part, you can say 30% of the cases whether it's familial or sporadic, 30% are explainable or genetic mutations are seen in 30% of the cases. That means rest 70% still we don't know what is the reason that they are getting the they have the Parkinson's disease. There are it is unknown, either because most genetic studies are required or maybe some other factors that we need, environmental or other factors. Age is naturally a risk factor and other factors are there. And in the recent time in India especially, so a lot of young onset Parkinson cases are seen. Young onset means the disease will manifest before age of 50 years. Now there are some genetic basis to it. For example, genetic mutations in certain genes such as SNCA, LRRK2, PINK1, etc. Mutations in those genes are more seen in the young onset Parkinson's disease cases compared to older onset. Older onset, for example, Parkinson gene mutations are common in the older, above 60 years PD cases. Now, genetics is not only important in diagnostics or screening, but also, pharmacological decision making, and because the drugs that are used to manage the disease, there is no cure for Parkinson's disease, but the drugs used to manage or to minimize the symptoms. So, the genetic mutations have a role in the effect. There are clinical genetic studies that have shown, like people with certain mutations or certain drugs are more working better compared to the others. So not only in terms of drugs, but for example the advanced treatment like deep brain stimulation. So the outcome of DBS also has genetic mutations in certain genes has a role in the outcome of the DBS as well. Now, the good part is in the last 5 to 10 years, lot of research, genetic research has been done. Also from India, 2-3 very good papers have been published. So on Parkinson's disease cases and controls, and which shows that how the more clearer picture about the genetic basis of Parkinson's disease, especially in India or South Asia. Now, fortunately, we have the test available, screening test available. So if somebody is having a, family history of Parkinson's disease, they can take a test, at 18 or 20 or 30s. So much before any clinical symptoms start appearing. So as you know, the genetic makeup remains constant. So one can take this screening test to assess themselves to whether they will be having their genetics is having a higher risk of developing Parkinson's disease at the later stage, and then they can try consult a clinician and try to mitigate the risk. If they are falling into higher risk, they can mitigate the risk of developing the disease in future. So this is much before the genetic test can be done, as I was mentioning, much before any clinical symptoms start appearing. And these are nowadays, as the technologies advance, these are very affordable tests that are available. Thank you.